Eugenio.
Eugenio, tranquilízate un
Estamos
poco.
en el coto, bueno.
Hay una pregunta en el chat.
Seguir la TAM.
Oigan, no, ya estamos en vivo en YouTube, ya, sean serios.
Ya, hay que admitir.
Ya estamos en vivo, chicos. Estamos en vivo.
Ya voy a admitir.
Oigan, me avisan si hay gente del extranjero, si no, para que lo hagamos en español.
Va a estar en español.
Ok.
Te avisamos, te avisamos.
Hay alguien de Brasil, puedes hablar en portugués si
Pueden
quieres.
checar si desde YouTube pueden compartir la
Preguntado.
opción de compartir el en vivo en
Ya, se
Facebook.
están yendo, güey, se están yendo. Ya voy a aceptar.
Güey, dices
Ya,
preguntado.
Eugenio.
Cualquier cosa es preguntado.
Cualquier cosa dices, mátale a eso. Verde. Sí, YouTube. Ay, se me olvidó.
Chicos, estamos en vivo en YouTube.
Cállate. Cállate, Sofía.
Oye, Ryosota, el japonés de Bonnie.
Güey.
Entonces va a ser en inglés.
Sí.
Hello, everyone.
Hello, everyone. Welcome to this Public Journal Club session. We will be starting in a few minutes. Today, we will discuss the paper by Kazuko Honcho Lab, which is RNA-binding motifs of hnRNA-PK are critical for induction of antibody diversification by activation-induced cytidine deaminase. This paper was published in PNAS this year, so I hope you enjoyed it, and we are ready for the discussion.
This is actually a very unusual occasion in which the authors are not going to join, but we are going to try to make it a very interactive public journal club this time. Rio.
Hi, Rio. Can you hear us?
Okay, so we'll give 2 more minutes for people to join, and we will start. So for the people who don't know, Public Journal Club is a project that we started due to this quarantine, which is the idea is that this is an open forum for scientific discussion, and every week we invite, we discuss a paper, we select an immunology paper, we invite the first author or the last author, and that way we can, after discussing the paper, we can have a session of questions and answers with the people who created the paper. So that way, it's a very unique opportunity for us as students and people interested in science to discuss and engage in a very interesting discussion with the authors. However, on this occasion, unfortunately, the first author that was invited had a problem and will not be able to join. In any case, we hope we believe Public Journal Club is more about the discussion among us, so we will be having lots of discussion this time. So we will start now. In this occasion, as I said before, we will discuss the paper RNA-binding motifs of hnRNP are critical for induction of antibody diversification by activation-induced cytidine deaminase. This paper was published by Tasuku Honjo Lab. The first author, Siwei In, will not be able to join, but we will discuss this paper that will be presented by Emmanuel Flores Espinosa. Emmanuel did his bachelor studies in basic biomedical research at UNAM, at the National Autonomous University of Mexico, and where he has specialized in signal transduction and immunology. So Emmanuel has participated in a member of the Public Journal Club, and it's also part of the board for the Bachelor in Biomedical Research. So welcome, Emmanuel. Whenever you want to start your presentation.
Thanks, Diego. Eugenio, ¿estás compartiendo pantalla? So, Eugenio, can you stop to... Thanks. So thank you guys thank you, Diego, for the introduction. And well, as Diego said, today we're going to discuss this paper that was published at Proceedings of the National Academy of Science of the United States of America, PNAs. RNA-binding motifs of hnRNP are critical for induction of antibody diversification and activation-induced cytidine deaminase. This paper was published by Honjo Lab, and here you can watch what are the members of this lab. So this paper is focused in a process that is occurring in B cells. For as all of you knows, and also by the class of the last Wednesday, B cell is a cell of adaptive immunity, and it's a commitment to principally to antibody production and well, the development of the B cell is through diverse is in start in the bone marrow when progenitor B cells are differentiated to pre-B cells, immature B cells that are characterized by the expression of IgM, immunoglobulin, and immunoglobulins are proteins that are that can detect antibodies and can exert diverse immune features. Principally, these immune features are classified as humoral immunity. So, mature B cells are able to be induced in an immunologic stimuli. So, when it happens, mature B cells goes into a differentiation program induced by stimulation by antigen presentation or antigen encounter, as says here, and also by cytokine signals that induce this stadium to be blood and later in the follicle in the follicles where B cells are grouped, they are a synapse is produced between T cells and B cells, and this produces that B cells can differentiate into memory B cells or into long-living plasma cells. But two process occurs at these centers and follicle centers. Well, in this, there are that are SHM and SSR. SHM is somatic hypermutation, and CSR is a class switch recombination, and we are going to talk more about this. So, somatic hypermutation and class switch recombination are two processes that are very related, and we can they are commitment to produce specific antibodies structure. An antibody, as you can surely you saw it before, has this structure. This is the general antibody structure when you have heavy chains, light chains, and you have constant regions and variable regions. So in these two regions occurs the effect that I gonna talk about it. Somatic hypermutations occur at the variable region where there is this is the gene of the immunoglobulins, the locus of immunoglobulins, and we can see here three regions that are named like B, D, and J. These regions suffer recombination by this process of somatic hypermutation that induce rearrangements of these locus. So every single process of somatic hypermutation conduces to a different structure and the different ability to recognize different antigens. So this process is part of the tools that permit that the immunological system or principally the antibodies recognize many antigens are even when it may be any antigen present in the universe. So it's a very interesting process. And also a class switch recombination occurs in the constant region. And this region, you have a set of different clusters that has the sequence specific that is specified for one class. We have different classes, CH, class D, G, E, and A, that are principally they are named by its Greek letters. But they produce a different antigen class. So how this process works is that it's by the action of one enzyme that we are going to talk, that is 8. Its activation induces cytidine deaminase. This enzyme it is function like it's an enzyme that induces cutoffs on these sites that I'm marking. They are known alike as regions. When the 8 is recruited to this locus, it cuts in different sites depending on the cytokines secreted in the follicles by the T follicular helper cells. For example, if there is secreted interferon gamma, the rearrangement will produce IgG. If IL-4, IgE, and if there is also DGF beta, it will produce IgA. And how it happens? It happens depending of in which side the cut is happening. The first cut is always here. You can say here first cut, but depending where is the second cut occurring, the cell will produce a different class. So if you cut here, you can eliminate this, and you will have the immediately next region. So you will produce IgG. But if you cut here, you eliminate also IgG, and you produce IgE. And if you cut if the 8 cut here, you eliminate IgE region and you produce IgA region. So this is how by all these steps are produced the different antibodies. So these two processes permits the diverse repertoire of antibodies. Sorry. Next, sorry. Next, we are going to talk in this paper about 8. 8, as I say, is activation induced acidity deaminase. That this his name is because this enzyme change the cytidine in the DNA or RNA. It changed cytidine by uracil. So this by the deamination of the cytidine, these bases are changed to uracil. So the action of 8 is to produce DNA bricks and adding the effect of deamination. It's a very complex enzyme, and its enzyme interacts with another RNAs and with another proteins that are regulating its activity. So about the mechanism of 8 action to produce the different cuts in the immunoglobulin locus, there is 2 hypotheses. The first one is that DNA produces a deamination. DNA is deaminated by this enzyme. That is what I showed here. That is the most is one of the most comproved hypotheses, that the action of this enzyme is directly to induce deamination in these sites, and that the change of cytidine to uracil produce this accumulation of the base pair, and it induces the DNA breaks, and single break first, then double break, and it recludes the DNA repair machine. So the second one hypothesis. The second one hypothesis is that, well, we have a question, Jacqueline.
I think they are asking about the like if everyone speaks Spanish, but if someone speaks English, we can continue the discussion in English. So please, if yeah, just continue, Emmanuel, and if someone speaks English, please write it on the chat. Thank you.
Well, I continue. If nobody speaks English, well, I can change to Spanish. So the second one hypothesis is that this enzyme deaminate RNA. Can you, as you can see, RNA has also cytidine. So what cytidine is deaminated is the problem here. And also RNA cytidine could be deaminated to produce uracil. And this second hypothesis says that this deamination is editing the RNAs. So these RNAs edited produce the DNA, produce or are important for producing the DNA breaks and repair. As you can see, the second one hypothesis is more complex. And it needs another proteins, and by the action of RNAs, these proteins can be ribonucleoproteins, and just that was there is more so much evidence that ribonucleoproteins are participating in this process, and a very specific ribonucleoprotein is this one, hnRNP. This enzyme.
Diego, sorry, Emmanuel.
What happened?
Diego, your mic is okay.
Well,
Sorry.
hnRNP. Yeah.
Yeah, Emmanuel, go ahead.
Thank you. Well, this is a poly-C binding protein, so it can be recruited to where there is many cytidines in one locus. So there was observed that in another studies that this protein, is specifically necessary for age-dependent DNA breaks. So here, Honjo Lab was interested in first in how is the interaction between hnRNP and AIDS, and how this interaction could be important for the role of AIDS-induced DNA breaks. So in order to respond that questions, in this paper we're going to see that they induce AIDS in B cells, in one cell line of B cells, that if you put in the media CD40 ligand, IL4, and DGF beta, you can induce the AIDS expression these B cells. And well, this is important for the rest of the article. Next, the class switch recombinations rescue assay is also important, an important method for this in this paper, that it consists that you have B cells expressing this hnRNP endogenous protein, And to this protein, you put one siRNA that blocks the expression of the translation of this ribonuclear protein. And but you complement with wild type or mutant versions of these proteins encoded in one plasmid. So you silence the endogenous expression of this protein, but you then you can rescue, that's the name rescue, rescue the phenotype by adding this topically this protein by adding one plasmid, and you can express wild type versions of this protein of a specific mutants of this protein. After that, you put a cocktail of cytokines, you induce the aid expression, and then you can see the effects. And the effects measured by them principally were IgA measure, IgA expression. They measure the IgA expression. So I see that any one person asked for the main question of this article. The main question of this article was how hnRNPK is interacting with A, and which part of this protein is important for this interaction, and if you can, if you can alter these key components to maintain this interaction, what happens in the cell? What happens when you don't have hnRNP? And then you can, by this method, you can know what is the importance of these two components. So first, is anyone have a question? More questions?
Yeah, I guess Eugenio have a question. You can read also through the chat, but is why they didn't did CRISPR knockouts mutants instance of microRNA microRNAs, interference RNA.
Oh, well, very good question. So by this method, they want to rescue in one moment of the develop of the in one moment of the cell that is previous the induction of the stimulations to produce the hypermutation and the class switch recombination. So they don't silence this gene by CRISPR because they have these tools, and also well, I don't know, but I think that it's by not interfering with the correct development of these cells. So well, there's no so much development because it's a cell line, but maybe genetic instability, I don't know. So, well, in the first figure, we can see the constructs the first constructs that they produce in order to eliminate the motifs present in these clusters that are named KH1, KH2, and KH3. These clusters have motifs that these motifs present in this structure, and these motifs are found to be important for RNA interaction of this protein. So they want to mutate the key motifs of this protein to see what happens when the cell loses these sites of RNA binding. So they mutated these motifs from GXXG present in this pre-cluster to GDDG, and the modern one is mutated this one, the motif present in this cluster. Next one is in this cluster. The next mutant is this one, and they also produce double mutants and a triple mutant. So next they have this construction. They planned this assay of in one moment you induce you can put the plasmid and the sirna in these cells as we saw, and 24 hours later you induce with the cytokines the expression of A and the activation of the B cells to induce the hypermutation, somatic hypermutation, and also the class switch recombination. 24 hours they did Western blot, and 24 hours later they did flow cytometry. So first the Western blot was to check if if this mutant is changed the expression levels of hnRNPK. So they saw that there is no change in the levels of this protein in the different mutants, and it's similar when the levels of the wild type construction. They also put the wild type construction in order to rescue the most completely possible the function of this protein. So then they want to check if the IgA levels are changed by these mutations, and we can observe that yes, the wild type rescue the levels of IgE are rescued to 22.5, and you have when they prove the different mutants, we can see that there is a less level, very less level. This is produced a reduction of the first one as nearly the half of the levels of the one type reconstitution.
Manuel?
And you in the double mutants, you can see that when you have two motifs mutated, you can reduce even more the secretion of IgA. Yes.
Sorry, there's a question in the chat. Ali Hines asks, what happens with hnRNPK basal expression in the mutants?
The basal expression, you can see it here in the sirna control when they put megalovirus and you put the cytokines in this one, you can see that these levels. So
no, I think it would work if you go back to the previous figure. I think. Yeah, so
the levels of the protein.
Yeah, the levels of the protein.
Protein expression. Okay. So well, they don't show it, the basal levels. You have the no plasmid when you not well you.
But you need activation, right?
I confounding this. That question is responded by this one. I don't know if you can say my mouse. This one is the basal expression of this protein, because you don't have a reduction of the expression of this protein by the sirna, and you don't have the plasmid, another plasmid adding next the sequence for this protein. So you have here the endogenous.
Okay. Thank you.
You're welcome. And well, the importance of this the measure of IgA is that IgA is produced only when class switch recombination occurs. So if you have IgA, it's indicative that you have class switch recombination. We can remember that B cells first are producing IgM, that is the basal expression. But when the cells are activated and by the cytokine and the A is expressed, and then class switch recombination occurs, you have IgA. But when you have these mutants, you don't have the same levels of IgA as you can have here when you have the wild type version of this protein. So this is an important observation. And you can see clearly here, you have the different mutants. So the first there are three are mutants, simple mutants. The two, these two are a double mutant, and this one is triple mutant. And well, you can see that there is difference between the mutant versions and the wild type version. And so in summary, we can say that mutants of GXXG motifs decrease CSR from class switch recombination from IgN to IgA. And also, we can see that there is possible additive effect of the number of GXXG mutations. Like we can see that where you have more mutations, you have less IgA levels. So they also there is also in this protein, you can see here, the regions of this, the sequence of this protein. You have this protein has a region that is named Ki. This region Ki has four motifs that are RGG that are also important for RNA binding of this protein by this protein. So they can, as they say, that, so that the levels here, when you have the triple mutant, there is no the same level when you don't have any expression of this protein. They want to know if this other cluster is also participating in the RNA binding effects, and if these RNA binding effects are participating in the induced class switch recombination. So they have they do these mutants for this cluster Ki, these RGG motifs. So they name the motifs the mutant 6, 7, and 8, where they only mutate this cluster, but they also mutate both type of clusters. Here we can see that they mutate the different RGGs, but they also have mutated one of the GXXG motifs present in the KH clusters. So they did these constructs, and they evaluated what first if the expression levels of this protein is not changed between the different mutation, and we can see that no, but in the mutant 9 there I can see more expression, clearly more expression of this protein. Yeah, thanks Diego for doing that declaration. As I said before, IgA is the method by they by they detect the class switch recombination. So they are evaluated in these mutants also the IgA levels in these mutants. So when you have the Cerna control and you induce this you don't silence the expression of this the endogenous RNP. You have these levels by induction of the B cells 17.3, but you can see that this level of IgA expression is less in this mutant. So mutation of this of the RGG decrease the IgE levels, but not only the RGG mutants, also the RGG plus GXXG mutants that are when you mutate the two type of clusters. So when you aggregate these two, you have diminished expression and secretion of IgA that they detect by flow cytometry. And but one interesting observation is that when you have this mutant that is the mutant for the GXXG and mutant for the RGG, that is this one, you can see that these levels of expression of IgE are not the same that you have when you have abrogated expression of this protein. So again, we can see that when you eliminate these two types of RNA binding motifs, you have some levels of IgA production. So they say that it could be happening for the action of other proteins or for other motifs that are not known. So in summary, we know that the RGGs participate in class switch recombination induction by this protein, this ribonucleoprotein protein, and also that there is residual class switch recombination rescue activity in the double mutant.
Emmanuel, there's another question in the chat. Anvit Rajnath asked us if they have a control in the paper for a mutation that doesn't change the IgA levels. And I think that's very interesting because all the mutations are any other mutation that is not affecting IgA levels.
Yeah, not all these type of these motifs are participating, no. So it's interesting.
Yeah, but I think
maybe there is a redundance.
The question would be like, if all the mutants, no matter what you do, if you do any mutation, you can get a decrease in the IgA levels, or is it specific?
Well, maybe I think that there is no specificity because you can see that there's very similar when you have 1 or 2 or 3 mutated cluster. So maybe it's the additive role of these different motifs that are producing the effect. But I don't think that there is a specificity by one of those.
Yes, I guess that what's it. Sorry, but I guess that what's said before through the chat, Abin Rajnath, is a pretty good question because it's the mutation per se, even before that specificity of changes in these motifs through the constant region of immunoglobulin. So I guess it could be given that the mutation per se, and there is no specificity, and this could be also a clue that why they did just a qPCR and instead to do a RNA-seq analysis, because they can evidence that they can see a specificity in these motifs regions.
Thank you. What do you think, guys? Anyone want to say his opinion about this question?
Yeah, I think it's a very good point, and I think they lack that control in the paper.
Yeah, well, Carlos, one person says, if I can elaborate more about this mutant, Mu6 is this mutant. Is mutant in the third cluster of KH3. No, sorry, in the cluster of the KI, they eliminate this they mutate this double RGG that is in this region of the 2096 amino acid. They mutated to ADD. So well, they have this effect that is similar to the another one mutants. So also then they want to see the effect not only because A participates not only in the class switch recombination, but A also participates in the somatic hypermutation. They want to know if there is an effect also in the somatic hypermutation. So they designed this experiment where they put the plasmid and the sirna to silence the expression of this protein and to complement the expression with the mutants. Then the cytokine supplementation, and then 3 days after the start of this experiment, they did a flow cytometry to check the IgA levels in order for a control, and also they evaluate the somatic hypermutation. You can see here the locus where they evaluated the somatic hypermutation. What was in this locus, the I and the C mu elements. You have the core as a new element when this core S is one side for binding of 8. So they evaluated if this region is hypermutated. Or there
We
is
have another 2 questions. Mark Bayer wants to know if both motifs, I guess the GDDG and the AAD, sorry, the GXXG and the RGG are RNA binding.
sorry.
Yeah, the question is if both motifs are RNA binding. I guess it's talking about the GXXG and RGG.
Okay, what was the question about these two?
If
motifs?
these 2 motifs are binding RNA.
Well, they don't evaluate this, and this is one of the elements that were absent in this paper. They only evaluated directly the binding or not binding of this RNA. They suppose that by mutating these elements, they don't have RNA. They maybe don't have RNA binding. And then in the next figures, we will see what insights about that point, but there was not direct measure of RNA binding.
Yeah, and Jesus Benito asks in another question if these motifs are necessary for the interaction of hnRNP with AID. And why did they choose to mutate specifically these sequences?
Yeah, because its sequence was found to be important for RNA binding, and also well, I don't know the previous evidence. No, well, the previous well, also GXXG is a motif that is important for localization of this protein to cytidine. So maybe they think that this is important for the AID action, because AID acts the amination activity is over cytidine. Is on cytidine. I don't know, but why they select specifically the RGGs, but maybe they have they identify it clearly these regions, and they don't have another regions of RNA binding specifically identified. What is the question?
Yeah, like if
What's a motif? What is important for physiology? Well, a motif of RNA is a sequence that is present along the open reading frame of along the sequence that is repeated. You along the more about along the genome is repeated, and there is a repeated in clusters on a specific localization, or there are located beside another identified regions, and the people propose that maybe these 3, 2, 4, 5 letters that are composing a motif is important for regulation. Thank you, Juan. So I will continue. Well, they so for the Western blot by Western blot the protein expression levels of these mutants, and they saw that there is the nearly the same level of expression, but you can see that there is a difference in the molecular weight of these mutants. They saw in the supplementaries by electrophoresis that there is a difference indeed in the molecular weight by by the different height of the bands founded by in these mutants. So they compare when they induce the expression of the aid and when they don't induce, and they saw difference only in aid, but there is no difference in the expression of this ribonucleoprotein protein. But when they evaluate IgA levels, they saw that there is also an effect. There is they confirm the phenotype that there is less IgA when you have these mutants. These two was previously checked, and this one is the double for RGXXG and RGG. And you can see that there is a reduction that is not the same as the wild type. So well, the importance of this figure is this experiment where they did an evaluation of the hypermutation, somatic hypermutation. They here they measure the mutation frequency. This mutation frequency here is showed. What about the cytometry flow? What you can have? What you want to see? Can you ask? Sorry, who wants to?
Maybe you can describe a bit what we are seeing, Emma, just to
Well,
see.
we are seeing the same as the last the past figure. We are seeing the IgA expression of these and these plots. Well, that they are the depicted here in the boxes, and you can see the number of the percent of expression of the frequency of these B cells positive for IgA. So if you have more cells expressing IgA, this number will be bigger, and the opposite. You're welcome. So they measure the mutation by the mutation frequency. This was detected by next generation sequencing, and they saw by specific regions in this one, that is this 447 base pair region. They saw that they saw the different SNPs and different sequence in that specific region. So we can, here is the depicted the mutation frequency that is in the wild type, and when you induce the 8, and when you don't have 8, that is the control. When you don't have 8, you don't expect to have so much differences about your reads, your different reads in the different cells. So they found a more diversity by the permutation here in the wild type condition, but when they induce the expression of A, they don't see the same levels of mutation of mutation frequency in the mutants of GXXG and RGG. Then they check for this difference between the cytokine supplemented condition and when they don't put cytokines in the media. And this difference is depicted here, and you can name this as delta mutation frequency, and you can see that this change is more here in the wild type, but this is decreased in the mutant conditions. Also, they so by the mutant frequency ratio, that is the wild type is is the 100%, and the no plasmid condition is the 0. Is assigned as 0, and they you can compare between every of this one, and you can see that there is indeed less frequency in comparison with the mutant, with the wild type condition. Sorry. So they have these two in summary, there is deficient somatic hypermutation rescue efficiency in these two mutants. So they have that these two motifs are important for the two phenomena that are important in this or that are for the B cell physiology, that are class switch recombination and hypermutation, somatic hypermutation, that produce diversity in the antibody. So when you don't have these motifs, or you can see that indeed these two processes are affected. So what is happening? What molecular mechanism? What could be the explanation for why there is less somatic hypermutation of class switch recombination? Well, they did. They want to know about what is happening about DNA breaks. DNA breaks are important for is one of the steps for both process for the action of A, that is class switch recombination and hypermutation. So they want to to measure the levels of DNA breaks. So first, we can see here the how was the system. They have it's known that there is translocations between the CMYC locus and the IGH locus. So there is an specifically the exon 1 of CMYC is translocates to the chromosome where is the locus of IGH, and this is happening in the cells when 8 is active, because this capacity of 8 to induce double breaks induce more this effect of CMYC translocation. So measuring the CMYC translocation is one of the strategies that they use to measure that indeed there is DNA breaks, because you don't can have a translocation of one locus to another if you don't have DNA breaks. You have to break one the change of DNA in this region to the sequence can be translocated to another locus. So they planned this experiment when they put the siRNA and the plasmid that we know, but also a siRNA for topoisomerase 1. This topoisomerase is impeding in some levels the activity of the action of translocation. So when they want to silence this topoisomerase in order to induce this translocation in levels that they can evaluate the difference between the mutants. So next they put the cytokines to induce the A8 enzyme. They did Western blot and also flow cytometry to check also that there is a reduction of IgA, like a control of and the principle is that principally they measure translocation. So, and in C we can see the picture of southern blot experiments for translocated CMYC. You have they did two experiments where they have the different versions of the hnRNP wild type and the different different mutants about GXXG motifs, about the RGG motifs and the double. And we can see that indeed there is more translocation by defined by more a signal of southern blot. They use one oligonucleotide specific for this region, that is nearly the exon 1. So they by this experiment in the southern blot, they detect the DNA region of this exon 1 present in the locus of the immunoglobulins. So if there is the signal of southern blot, there is that is a signal for if hypermutation. So for translocation. So we can see that there is indeed translocation in the wild type condition, but this translocation is diminished in the different mutant conditions. So also the translocation is affected in these mutants, and maybe it's not the direct measure, but that maybe that could be by in decreased DNA breaks.
Emmanuel, they are asking if why is the exon 1 upside down in the figure A?
Well, because the 5 ¿Cómo se dice prima? Well, because it translocates in this one, the five
prime.
5 prime is translocated to this side, and the 3 prime extreme is here. So there is the translocation is when you have one segment of one chromosome is inserted in another chromosome, but is in the is in the in the opposite direction. Yeah. So then they check
Emma?
the translocation frequency. Yeah.
Sorry for not asking this in the chat, but I'm in the cell phone. It takes me forever to write. But I don't know. What are your thoughts in Figure C? Because it seems to me that in the wild type, not only you see this increase in translocation compared with the mutants, but it seems to me that the molecular weight of the bands are also like more equal than in the mutants. I mean, it seems to me that the translocations are more like stochastical in the mutants compared to the wild type. And I don't know if they mention anything about that in the article.
No, they didn't mention if there is a specific translocations or if there is a different translocations by. I think that maybe it's by differences in the different B cells that are present in the population. I don't know, but as you can see that the don't translocates in the exact same site in every translocation event. No, the difference in the molecular weight, and also you as you check there is difference in the strength of the in the mark.
Okay, thank you.
You're welcome. Well, then they check the translocation frequency, measure by measuring in graphs that was showed in here in C. You can see measured here indeed. You in the wild type conditions, you have more translocation when you induce when you put the cytokines. And these levels of translocations are not recapitulated by the different mutants. Well, there is no statistical deviation, because they are depicting here only the one of what is showing here. They don't I don't know why don't show the mean of the different experiments. They are only they say that this was not the only one experiment. They do the well, they're showing here 2 experiments, and they did what is the in.
Emma, do you know what are the number of in parentheses? It's the number of
the number in the parentheses is the number of different what is the name?
I think it was the number of cells with translocations, or
no, it's the number of different lines, the different carriles.
the lanes, yeah.
That are that have a translocation. So there is more
Thank you.
translocations here than in other. Well, in E, we can see they want to measure more directly the DNA breaks. So in order to measure that, they did they planned this experiment where they put the plasmid and the serum that they use in this experiment. They induced with cytokines, and 24 hours after they did the LMPCR and double a DNA break assay. So they show here that where is the proof for the locus that is in this region in the score of the mu site for 8. So they can detect this near the site where 8 is it can be recruited. So it's in this region is probably that there is a DNA breaks caused by 8 specifically. So but they amplified these regions about the cells, the B cells, and this time point. And in the when you have the what in the that RNA was they did a southern blot, and they put a probe for that region, and we can see that there is more fragments of DNA in the wild type conditions, and there is indicative that there is more DNA breaks in that region. But in the mutant conditions, you can see that there is less. Even here is very little DNA breaks, and you can see also that clearly that DNA breaks are diminished. So the mutants are the 3 mutants, the 3G that are all X GXXG sites mutated, the 5 ADD mutations that are the 5 sites RGG mutated, and the double mutation of that have reduced translocation frequency and also have less DNA breaks. Well, they another one question is that hnRNP binding to RNA is important for its interaction with 8. So what is happening when you don't have that motif in the hnRNP? How is the interaction between hnRNP and 8? Well, they wanted to evaluate that, and they did this experiment. They have the plasmid and the serum put it in the cells to do the reconstitution with the mutants. The cytokines, they did immunoprecipitation 24 hours later, and also 24 hours later, 3 days after the start of the experiment, they check for the flow cytometry, the IgA expression. So we can see here they evaluated GXXG and the RGG mutants. They co-immunoprecipitated by co-immunoprecipitation, they want to check the interaction between 8 and hnRNP. By immunoprecipitation, you can precipitate one pair of proteins, and you can detect with an antibody for the another one protein that is interacting in that pair of proteins. So here is the input of the here is the input of the samples of the experiment, and we can see in the input when you don't precipitate that there is the same levels of the hnRNPK protein between the different conditions of mutants and wild type. This one is a control when they put the serum, but they don't. They put a plasmid for cytomegalovirus or virus, so they don't have. They have the serum residue levels of residual levels of hnRNP. So they also call it M8. It's not affected, but in the immune precipitation, they have the same levels also of this protein, but there is not the same levels of interacting with hnRNP between the mutant conditions and the wild type conditions. The line that is above here is marked with an asterisk because this was found to be an inspecific, but maybe because the antibody is in a specific band. So please, if you can see only this one that is of 24 kilodaltons, you can see that there is more expression of in the wild type condition, and there is less levels of M8 interacting with the precipitated hnRNP. So this was with GXXG mutants, but this also happens with the RGG mutants. You have less levels of interaction of 8 with hnRNP when you have the mutant conditions that was when you have the wild type version of the protein. So here indeed they did the same experiment, but with different mutants. They have the mutants that are all the same type of motifs mutated, all the same type of RGG motifs mutated, all the motifs are for GXXG, and all the motifs, all of the motifs of CXXG, and also the RGG here in the center. So we can see that also here the levels of interaction of M8 where the ribonucleic protein are less in the mutants than in the wild type, but well, you can see maybe here is less protein of hnRNP, but we can see also that again that the different mutants induce a little of difference in the molecular weight of the proteins. After they measured that signal of that Western blot about the immunoprecipitation assays, and they about different experiments they found the average of signal of immunoprecipitation and was normalized by the wild type condition. In the wild type condition is 100, and we can see that the difference in the wild type conditions about the input and the immunoprecipitated 8 is the same. So it's interesting, but this indicates that all the all the 8 present in the cell is interacting with hnRNP. With I don't know if this could be true, but they found that it's the same. So it indicates that all the 8 is always interact with this ribonucleoprotein, and maybe this ribonucleoprotein it's another indicator that this is very important for the 8 action. So in the mutants, you don't see that. You can see that immunoprecipitated 8 with that hnRNP is less. You have less, and the mutant one is the half, but you can see here in the mutant 7 is very decreased interaction. And well, there's difference. There's no significant difference between these. But there is difference. So in summary, the GXX and the RGG mutation decrease the ability of this protein of hnRNP to interact with A. So also we can see that these motifs are important for the action of A and also indeed for the interaction between.
Emma, before you continue, Sarah Holick was asking if a motif means a function.
No, not always.
He said, I
Not
think that
always.
distinction is important. Why not just saying binding of a protein? Why the motif concept?
Motifs are more genome analysis concept that you have a structure, you have a, what can I say? You have conserved elements about along the genome, but there is no a direct relation about the one motif has a specific action or a specific effect in the cell physiology or in the cell activity. No, it's always that in that order. Anyone can say it. Yeah, you're welcome. GXXG and RGG motifs were also evaluated for the RNA binding capacity of hnRNP. So that this could respond answer the question of, that was asked about if indeed these motifs are important for if they measure the RNA capacity of this of these motifs. Well, they did this experiment when they want to evaluate by RNA immune precipitation, like that, as Carlos say the last Wednesday. RNA immunoprecipitation is you have an RNA that maybe is interacting with one protein. So if you want to know if one protein and specifically is interacting with RNA, you put an antibody specific to that protein. You immune precipitate that sample, and you, by different methods, can detect the RNAs interacting with that protein immune precipitated. So here they want to evaluate these RNAs that in the literature was founded that these RNAs can interact with the hnRNPK protein. So they the experiment in the immunoprecipitated with antibodies about the against the hnRNP protein in the different conditions this protein, and they evaluated the percent of by qPCR. They evaluated the quantity of the RNA found to be interacting with the protein. So in the one-set conditions, we can see that indeed there is a important interaction with of this protein with semic, CARN1, and link this long RNA, and also with the different functional RNAs. But when you have the different mutations, it is interaction of this binding capacity with the different RNAs is dramatically decreased. So these motifs are important for RNA binding by this protein, and that is that maybe is important in the effects that they saw. So they detect the enrichment of two types of RNAs. That is the reported binding RNAs. That there's where there is evidence that RNAs are binding to hnRNP, and also they evaluate for the long non-coding RNAs that can be binding this protein. So about along the different RNAs that have a function of DNA breaks, we can see that the wild type can interact with these RNAs, but not the mutant. We can detect there is very little interaction with the we can see clearly, but there is a dramatic reduction in the binding capacity with this type of RNA that are implicated in DNA breaks, but also in the RNAs implicated in DNA repair. There is an effect. There is a decreased binding capacity, and also in the long non-coding RNAs, except for the need one too. This way is one was discussed in the paper that maybe it's because this mutation is altering the structure of the protein, and this could enhance, maybe this specific alteration can enhance the structure that is interacting with this RNA. So there is an more enrichment in the mutant than in the wild. But in the different that are not need, there is a reduction of the binding capacity, RNA binding capacity. Another effect is the localization of hnRNPK. The localization of this protein is dynamic. It's not always in the same compartment of the cell. There is, in the basal condition, this protein is present in the cytoplasm, but its activity is exerted in the nucleus. So it has to be located to the nucleus. So as you can think, this protein is produced in the cytoplasm and has to travel to the nucleus. So the localization in the nucleus is an indicative that this protein is maybe active and is maybe interacting with A. So we have the plasmid. They did the same experiment as always. Plasmid, sirna, adding the cytokines, and they evaluated 48 hours after the cytokine induction. They evaluated the protein localization in cytoplasm and nuclei and in the whole cell. That means W. So about the different mutants, we can see that there is less localization in the nuclei when we compare with the wild type condition. So all the mutants have less nuclear localization, and maybe is not clearly, but there is also some less nuclear localization of 8 in some mutants like this one or this one. So they use like a nuclear localization control the topoisomerase that is present in the nuclei, and for cytoplasm whole cell, the GAP-DH that is an enzyme present in not in the nuclei. So yeah, when we can see clearly that there is a difference in the localization. These mutants are not able to relocalize to the nuclei. So they want to know how this failure of nuclear localization, how is compared with when you mutate the nuclear localization signal endogenous of this protein. This protein to localize to the nuclei thanks to this signal that is a peptide sequence that is specific for nuclear localization, NLS. So they did this mutant when they changed these amino acids for alanines. So this nuclear localization sequence cannot work. So in the different mutants, they compare this mutant, but there's the mutant 12 with one of the mutants for the GXXG, and as is expected, this mutant for GXXG is not present in the nuclei, but the mutant for the nuclear localization signal is also affected, but not in the same extent. So maybe is that this nuclear localization sequence was not affected the completely, or maybe the role of these motifs are indeed so important for the nuclear localization, maybe even more than the nuclear localization signal. There are maybe these motifs are important for the nuclear localization in some extent that can induce the effects in the aid activity. So they put here in the numbers for if you want to compare more clearly about the levels of nuclear localization when you the nuclear localization, and also compare with the class switch recombination rescue. And well, this is the when you have all the wild type, you have all the class switch by normalization, but you have only 41 localization in the nuclei. But you can see that when you have the mutant for NLS, you have only 23% of the protein present in the nuclei, but you have the 90% of the effect in class switch. So maybe they're only necessary 23 or one little more of the protein present in the nucleus to exert this function of the hnRNP. So it's interesting. Finally, in the summary, we can in the paper they found that the wild type between the difference in the mutation of the cluster of the motifs H, XXG, and RGG compared with the wild type. When you mutate this protein, you have you decreases RNA binding, less nuclear retention at the cell level, less binding to the aid enzyme. But in what happens when do you have that? Well, in the molecular biology level, you have less DNA breaks, and in the physiological level, you have less class switch recombination, less somatic hypermutation, and less translocation. So this phenomena was evaluated, and we can see that one molecular component that are these motifs are important for the activity of one of the eight enzyme, and this is such important that if these components are not present, there is a phenotype. And you can see here there is less class switch recombination, less somatic hypermutation, and maybe there is less diversity in antibody produced. So they don't check, but surely they could be less. It could have less diversity in the antibodies. So that was all the paper. If anyone has questions, please say that.
Thank you, Emma, for this explanation. There's a question in the chat. I will read it. The GXXR mutants show that high nuclear translocation as and a less mutant, but CSR was so much lower. Nuclear retention cannot fully explain the efficacy of CSR. What do you
Okay,
think?
it's from Ryota. Okay, showed us high nuclear translocations. Well, we can see here that there is not the same extent. There is less nuclear translocation of this and of this protein. Here, there is less nuclear localization of this mutant when you have this mutant than when you have the nuclear localization signal, nuclear signal localization signal. So that was not what you asked. GXXR mutants showed as high nuclear translocation as R mutant. No, that was not the same extent. There is a more translocation of the mutant. Sorry, of the mutant for the motif, but yes, what class switch recombination was so much lower. Sorry, I will present again. Yeah, in this table we can see that that is lower the localization. Sorry, it's lower the rescue because you in the nuclear localization signal you don't have the you don't are eliminating the RNA binding capacity. So this is interesting because there is not only the localization to the nuclear, but also the RNA binding capacity of the protein important for the effects of AIDS. So that's the answer to your question. The RNA binding capacity is important. You're welcome.
Okay, anyone has any other question?
I have a question. Well, it's more like a comment because I don't know if I'm misunderstanding, but they say that one of the reasons of why they don't see RNA binding of the target RNAs with the mutants of H and RNAP is because the majority of the protein is not localized in the nucleus. And I was wondering if they did either in supplementary figures or if they explain anything in testing the binding of target RNAs to the different mutants, of the hnRNP, because they use in the figure, they only use the mutations of combined motifs, but not each motif separately. So I was wondering that because it seems that in all of the mutants, in the combinations of the motifs, they lost completely the binding of the RNA. And I was wondering if they did like each motif separately, they will see like binding of RNA differentially depending on the target. I don't know if I'm explaining myself. I'm just going around in circles.
Yeah, that was that they in the principle of the article they worked with the different motifs, no, individual, but later they don't work with that. Maybe it was by time and sources consumption. Maybe for imagine this, for example, this Western blot. Wow, how many these three conditions will have to be replicated in another one, 6 or 7 more mutants. So
Yeah, it would be a lot.
yeah, and also your question about the different RNA that maybe it's interacting. They even in the in the discussion of the article, they say that maybe is the next step is evaluate the which RNAs that are specifically interacting with hnRNPK, and also are identified like important for the H function are indeed interacting with this protein. If that RNAs continue interacting with the protein when the motifs are mutated. So
Yeah, because it will be also very interesting to know whether if each motif confers like specificity for different RNAs, like
yeah.
target RNAs, and they wouldn't mention anything about that.
Mhm.
And also maybe if they perform like an experiment trying to sequester the hnRNP maybe to the membrane or something to avoid it to enter to the nucleus, maybe they can dissect the difference between the importance of the motifs and then localization of the protein, right?
Right, and that's a very interesting possibility. Maybe in the if you don't put these alanines in the nuclear localization sequence, but you have put change this signal to no iron endoplasmic reticulum localization signal to sequestering the protein in another cell compartment could be interesting. What happened? No,
Yeah. Thank you, Emma.
you're welcome.
Any other comments or questions? All right, if there's no more questions, thank you again, Emma. It was a very nice presentation and a very nice paper. So just before you all go, could you please turn on your screens to take a picture, your cameras to take a picture for the record?